So how is the acid-base balance maintained in our body? We have first line defense and we have second line defense. So they are categorized according to the mode of action. So our body's buffer system, that is the chemical buffer system, they are going to act within minutes. So they are the bicarbonate, phosphate, protein and hemoglobin buffer system, which comprises the first line defense against the pH shift. So whenever there is more amount of acid or wherever there is more amount of the base produced, so these buffer system, they come in action and they try to maintain your body's pH. The second line defense is your lungs, which is within 1 day. So this lungs, they are the second line defense and they are acting longer than compared to your chemical buffer system. And then comes the third one, that is the renal mechanism, which is the permanent solution for the acid base balance, which gets which rises after, or which comes in action within 4 to 5 days. So this is the buffers, this is the body's defense system to combat the acid base abnormality in the body. Now moving on to how are we going to control this acid. So we have buffer system. How is this buffer system acting? So they are taking up more amount of the proton, that is H plus, which is released, which is coming from the foods or coming from your metabolic action, or they release H plus as conditions change. So whenever there is more amount of the alkalosis happening, so it releases H plus, which it is which it contains, or it mostly takes up H plus whenever there is acidic condition. So buffer pair is like that, that is it is a weak base or a strong acid, weak acid or a strong base, that or vice versa. So by this action, so whenever there is more amount of your suppose this is the carbonic acid, so they dissociate into H plus and your HCO3. So whenever there is more amount of your acidic environment or the body has less pH, this HCO3 is reabsorbed in a larger amount. So this maintains, or this tries to decrease the acidic burden in the body, that is it tries to maintain the pH or raise the pH, while this H plus which is there, it gets excreted. Now whenever we have more amount of HCO3, what does this do? This HCO3, they combines with H plus forming H2CO3, and this if according to the change in the body's pH, this CO2 it gets exhaled in the your exhaled outside, that is exhaled in the exhaled air, or by this carbonic acid, it contains H plus and HCO3. This H plus is tried to conserved in the body whenever we have increase in the pH. So in this way there is change, or there is a very minimal change in the pH whenever buffer come into the action. Now this is a tabular form which is showing the main buffers in the human body. We have hemoglobin, we have proteins, we have phosphate and we have bicarbonate. Among this, the major extracellular and your potent buffer is your bicarbonate buffer, and then comes your phosphate buffer, your protein, hemoglobin. These are mainly the most predominant intracellular buffer is your phosphate buffer, and then we have proteins which are present intracellularly. A small amount, that is less than 1%, is in extracellular, and hemoglobin which is present in the erythrocytes. So these are the components which are collectively known as the first line defense of the body system to maintain the acid-base balance. So now coming on to the most potent and the extracellular buffer, which is very important to maintain the body's acid-base physiology, is the bicarbonate buffer, which is made from the your combination of sodium bicarbonate and a weak acid, that is your carbonic acid. So this bicarbonate buffer, it accounts for the majority of the buffering capacity, that is 65% of the total buffering capacity. And if you see the values of your bicarbonate, carbonic acid and the pKa of your the bicarbonate, if you if you keep this in the formula for Henderson-Hasselbalch, we can find that your pH, that is the body pH, is 7.4, 6.1 is the pK, and at this concentration it is around it comes around 1.3. The pH of the pKa of bicarbonate, though it is 6.1, the concentration of bicarbonate is too high, that is the ratio is always 20:1, that is base by acid ratio is always 20:1. So which maintains the alkali reserve of the body as well, and this causes the quick maintenance of the acid base imbalance by your first line defense, which is the bicarbonate buffer. So we should be knowing about this bicarbonate buffer in detail because it is the most potent and the most effective buffer. So the second thing which is very important in the bicarbonate buffer is two organs are maintaining this bicarbonate buffer efficiency. The first one is the lungs, that is via formation of H2CO3, which breaks into CO2 and H2 in the further metabolism. This CO2 is exhaled if you have more amount of the acid concentration. If you have less amount of your H plus, this is always tried to conserve in the body. The second one is the renal, the erythrocytes, which is also maintaining your bicarbonate buffer, is also collaborating with your erythrocytes to some extent, and your kidney, your renal mechanism also maintains your is also affecting, or there is a direct effect to the bicarbonate buffer through the renal mechanism as well. So due to this reason, the bicarbonate is known as the potent buffer. The first one is the concentration, the second one is the interplay between the number of the organs, which maintains your bicarbonate buffer efficiency to be higher compared to the other buffers. The second one is the phosphate buffer, which is the potent intracellular buffer. Now I was talking about the pKa. The pKa of the phosphate buffer, if you see, it is much higher than your bicarbonate. It is 6.8, which is less than 6.1, and it is almost equivalent to your, or very near to 7.4, which is the body pH. So seeing at this, we can be having the idea that pH should be always more or less equal to pKa. That defines phosphate buffer to be the most potent buffer. But phosphate buffer is not considered to be the potent buffer because of its location. Since it is inside the cell, it is maintaining the pH inside the cell, and it has a pKa closer to the body's pH, but still it is not considered to be the potent buffer because of its location, and that's why phosphate buffer comes to be the second important buffer in the body. The another important point in phosphate buffer, if you see it forms dihydrogen phosphate and sodium hydrogen phosphate. Now these two are very important because they are forming the acidity, or they are comprising the titrate acid in the urine. So it is mainly the excreted acid in the urine, which is helpful for the which also helps the renal system to acidify the urine. Now the third important, or the collective buffers, the hemoglobin is comprised of, and we have a protein buffer. So protein buffers are very important intracellular buffer, and it comprises of less than 1% of the extracellular buffer. This protein buffers, why they are having the capacity of the buffer, is because they are comprised of different types of the amino acids, and because of the characteristic of the amino acids, this buffer can maintain the change in the pH. So they can give H plus, that is they have the capacity to protonate and deprotonate. So they can accept H plus or they can donate H plus according to the pH or according to the pKa of that amino acid, and due to this it renders this buffering this proteins to act as a buffer. So carboxyl group, since amino acids are comprised of your amino group as well as the carboxyl group. So what happens is this carboxyl group, they can give up, they are acidic, so they are H plus donor, and your amino group, they are basic, so they accept H plus or they can donate OH minus, and they contains different types of your amino acid. The most important one is the histidine. So histidine is present in major amount in your hemoglobin, and this histidine has your imidazole ring, which gives the potency of buffer to act as a buffer because it has the major function of protonation and deprotonation, that is accepting as well as donating proton according to the different type of or different pKa. So by this mechanism our buffering system maintains a slight change in the pH within minutes. Now coming on to the two important other organs which are the second line of defense, we have lungs and we have kidneys. So if you see that pH equals to pKa plus log base by acid. So here the first component, that is the base, it is handled by kidney and it is also known as the metabolic component. Then you have acid. So acid, it can be considered as H plus or it can also be considered as the carbon dioxide, which is ultimately giving the acidity to the body. So these are the respiratory component and they are maintained by the lungs. Now coming on to the second line of defense, that is respiratory regulation of pH, how it is done by the lungs. We all know that the lungs handles the inhalation and exhalation, that is it takes up oxygen, gives away the carbon dioxide, and in the same way it is maintaining the change in pH and acting as the acting in the acid base balance. So hemoglobin as a buffer, it is going to help or it is going to act through the lung pathway, and the imidazole ring which is present in because of the histidine, it is present in your large amount in your hemoglobin, which can accept or which can donate the H plus in the different pKa. So oxygen according to the different type of the pKa, it can donate as well it can accept the proton, which can hence resist the change in the pH. So if you can see that oxygenated hemoglobin, they are stronger acid, it means they can donate proton, and deoxyhemoglobin, they are acting as a base and they they takes up the proton. So 6.7 the hemoglobin is they are oxygenated hemoglobin and they can donate H plus, and at a basic pH it takes up H plus or it can give away the hydroxyl ion. So according to this mechanism, your hemoglobin acts as a potent buffer because of the potency given by the histidine for accepting and the donating proton, leading to the delivery of the oxygen as well as maintenance of the pH. So I'll show you a figure here. So this is the imidazole ring of the histidine, and at the pKa of 6, that is below a pH of the 6, it acts as a proton acceptor, that is it is mostly protonated, it takes up the proton. If you see here this is the imidazole ring and it takes up the proton, and at the pH above of 6 it gives away the proton, that is it becomes a proton donor or deprotonated, and because of this action, according to different type of the pH, hemoglobin acts as a buffer. So this is the major picture by which hemoglobin functions as a buffer. Since you can see here carbon dioxide which is produced in your tissue cells mainly by your metabolic action, so it enters into the through the interstitial fluid to the blood plasma within the capillary, and here this hemoglobin, they picks up your CO2 and H plus, so it becomes a deoxygenated hemoglobin, and when this deoxygenated hemoglobin they goes to the lung, here what we can see here is it gets because of this deoxygenated hemoglobin, it is able to take up the यो लास्ट हो, त्यसपछि है 250 भयो, तिमी निस्किप हाल्नुपर्ने होइन नि, यो पछि निस्किउँ न त, यति हो अनि छोड्दिउँ, रेनल मेकानिजम लामो छ कि है, म त ल्याउँछु जस्तो लाग्यो। So now here you can see that hemoglobin, they acts as a buffer by its structural ring which can donate and which can accept the proton according to the change in the pH, that is in a different pH it acts as a proton donor or acceptor. So here this picture shows that how is carbon dioxide is transported from the tissues, then it is that is by this mechanism hemoglobin maintains its takes up the proton so that the pH of the blood does not falls, and here carbon dioxide which is liberated, that is from the tissue cell which is produced, it diffuses into the capillary wall, it combines with water molecule by the help of carbonic anhydrase inside your red blood cell, this is the erythrocyte, it forms your carbonic acid. This carbonic acid is broken down into H plus and your bicarbonate. This bicarbonate, we all know that this mainly the bicarbonate is always reabsorbed in the body to maintain the alkali reserve. Here the another thing here is there is the formation of H plus, and the CO2 which is produced or which diffuses into the capillary wall, it is taken up by this hemoglobin, and this becomes a deoxygenated hemoglobin. Now after the production of the CO2 from the tissues and hemoglobin taking up those all extra H plus and proton and your CO2 to maintain the pH, that is it does not let the pH to drop, so it prevents the pH dropping. So after this, once it forms bicarbonate, it moves towards the lung. So this bicarbonate, it again combines with proton which is released by the hemoglobin, which here you can see the hemoglobin had taken a proton which was formed here so that it does not causes the change in the pH. It forms H2CO3, and depending upon what is the status of your body, if it is an acidic condition, this CO2 is taken in or exhaled in a large amount so that the body maintains the alkalinity or the neutral pH of 7.4. It is exhaled. If it is less amount of the proton or less amount of the acid is present, like in condition of alkalosis, this CO2 is always preserved, or it is not exhaled, or it is always there in the form of carbonic acid. By this mechanism, hemoglobin, it is going to take up the oxygen from the lungs to the tissues and takes up the protons and your carbon dioxide from the tissues to the lungs, and via lungs all this CO2 excess is exhaled. So by this mechanism, lungs is important in maintaining the acid base balance in the body. So this is the similar picture, which is in a very simplified form. We can see that from metabolic action we have CO2 and it combines with molecule of water forming H2CO3. This is inside the tissues and inside your erythrocytes. So this is the enzyme which is doing this is carbonic anhydrase, and this H2CO3, it breaks down into H plus and HCO3. At this point of time, you have hemoglobin, which is delivering oxygen from the lungs to the peripheral tissue. So this is the oxyhemoglobin, and this hemoglobin, after delivering the oxygen, it combines the H plus, which is liberated from your carbonic acid. This that is deoxygenated hemoglobin, is transported back to the lung. Along with this, we have bicarbonate also, which is transported, and this again combines with a molecule of water forming H2CO3, a molecule of proton, which is liberated from this deoxygenated hemoglobin, so that this hemoglobin can carry more and more oxygen again, and it is going back to the tissues. So this cycle goes on, and it is also known as the isohydric transport. Isohydric means at the same pH, at the same temperature, which is maintained for the flow of your that is the carbon dioxide metabolism is happening, and this is known as the isohydric transport. So this is the summary of what we learned, that is how is the pH of the body maintained via lungs. So whenever there is change in this pH, we know that normally we have normal pH 7.35 to 7.45. Some factors, for example, it is causing hypoventilation, that can lead to conservation of CO2. That is, CO2 is trying to retain in the body, which causes change in which causes decrease in pH, leading to acidosis. And if this decrease, that is conservation of CO2, is there, our baroreceptors and your receptor, that is the carotid receptors, are stimulated. So not the baroreceptor, baroreceptor. So this is the summary of how is the lung maintaining your body's pH. In case of hypoventilation, whenever there is more amount of your CO2 being accumulated in the body, it causes the trigger to the carotid receptors, which causes hyperventilation, and that causes more and more CO2 to be exhaled in the body, in the air, maintaining the pH to 7.35 to 7.45. If due to some factor which can causing hyperventilation, there is washing away of the CO2, this is leading to alkalosis. This alkalosis again, it trigger your receptors in the brain, causing conservation of the CO2 with hypoventilation, and that causes again conservation of CO2, so that H plus and HCO3 concentration is normalized, causing back to the normal pH. So in this way, lung is causing hyperventilation, and hypoventilation causing increase in CO2 exhalation or CO2 conservation, leading to the maintenance of the blood pH.